Hello and welcome to the Driscoll Lab. My name is Ryan Wasp. I'm a senior PhD student and today I'm going to be giving you a virtual tour. First, I just want to show you all the members of the lab on our main poster board outside. You can see all the people who work here. Here we have a news and events section where you can see all the events that are coming up that are related to biology and C. elegans here at Rutgers. And here we have our wonderful lab alumni who have been with us in the past, and we'd like to remember and look at them often. So to start, I want to show you some equipment and some of the rooms in the lab itself. This is the first room of 3 that I'm going to show you. Here we've got our brand new revolve scope. It's a wonderful microscope because you can actually remove the screen and move it around and take it wherever you want to. It's very mobile, and it's very good for teaching new people how to look at C. elegans, and wonderful for showing undergrads how to do worm work. This large recording setup allows us to take pictures and videos of worms either swimming or thrashing or crawling on a plate. And we can use some software that was developed here in the lab called Celeste to automatically calculate how many body bends a worm will do in different media, and we can measure different mutants, different stresses, and see how that changes using this computerized software. This wall here is our supply wall. We've got anything you could possibly want, from autoclave tips to snap cap tubes to mini prep kits, chem wipes, anything you need. This is our giant shared stock of equipment. Moving through the lab, you can see our fume hood where we dry plates and do a lot of work in a nice, clean, aseptic laminar flow hood. You can see various incubators and equipment. We have a brand new rack of freshly laundered lab coats, which is always nice. Moving into the next room, I want to show you guys our computer space. This is really nice. Anybody can use these computers at any time. They have all sorts of imaging software, statistical software. If you ever want to read a paper and print it out, you can do so right here. We actually have a working lab phone. This large piece of equipment here is a spectrofluorimeter. You can use wavelengths of light to analyze all the wells of a 96 well plate automatically, which is really nice for high throughput experiments. And here we have a bench, a nice bench. This is about as much space as everybody has in the lab, and this is a particularly clean and exemplary bench which belongs to Ricardo Laranjairo, one of the postdocs in the lab. Before I leave this area, I do want to give you guys a quick peek of our outside area. You can see that it's well manicured and it has some nice picnic tables if you want to eat lunch outside on a particularly nice day with your colleagues. That's an option. Very nice outside. Here we have some standard equipment. We've got a nice tabletop centrifuge, an incubator, and a stratolinker if you ever want to do some UV kill with bacteria experiments. This is our main PCR bank. We have 4 machines right here, but we have a few else in the lab as well. These are nice touchscreen machines, so they're really simple to use. And we do a lot of PCRs here, so it's nice that we have so many because you can always get time. This is a dedicated sink for our ethidium bromide waste. This is where we run our gels on this gel electrophoresis machine. We have several. Some people have some at their bench, but this is a dedicated area for that. And one other thing I'd like to show you is this glassware cabinet. What's really nice is we have a dedicated glass washer who will pick up any dirty glassware here. You just place it there, it'll get washed, cleaned, autoclaved, and replaced in the cabinet. So we always have a full supply of beakers, flasks, and graduated cylinders whenever you need it. Moving through the third room of our lab, you can see a pretty large array of incubators and refrigerators. This is basically dedicated entirely to the extrovert group within the lab. Everybody has shelf space. Everybody has a dedicated shelf, so nobody will ever mess with your stuff. It's all very clean and well kept and well organized. Just have a millicube machine here if you ever need to make media. And speaking of making media, here we have our chemical supply bench. You can see that we have a large array of standard lab chemicals. Whatever you need is generally here and shared between all lab members. We actually share this with another lab, but you can just take whatever basic supplies you need and you can weigh them out or adjust pH, or stir them, or we have a hazardous weigh station here too. But basically, whenever you need to make media, this is where you'll be making it, and everything is easily accessible and plentiful. We're a very eco-friendly lab too. This is our tip tray recycling center. We give these to a guy who picks them up, and they get melted down. I think they get turned into golf tees. So we're a very eco-friendly lab. Just thought I'd mention that too. So that represents the main lab, and I wanted to show you a few of our accessory rooms as well. We have a lot more space, and we have some interesting equipment that I'd like to point out as well. First, I just want to really quickly take a peek into the cold room. It's a bit loud in here. Along this wall, we keep our personal supplies, whatever plates we make, antibiotic plates, plates with specific drugs to test, we'll keep here. But I also want to point out that these boxes right here are full of NGM, which is made by a dedicated technician. Whenever you need to keep worms refreshed, you just take a big old box of pre-made plates, and you can see them yourself, and they'll be ready really quickly. I'm going to take you guys down the hall, but before we do that, I want to show you some of the beautiful posters that the postdocs and grad students have made and presented at various international conferences, such as the International C. elegans meeting in LA, which we attend often, as well as the Stress and Aging meeting held in Madison, Wisconsin, which we also attend when it's held. Unfortunately, it's canceled this year due to COVID-19. So I'm going to show you some other important equipment in this room. We have a transilluminator to check your ethidium bromide gels if you just need to really quickly see if it worked or it didn't work, if your PCR was successful. And here we have a brand new gel imaging system. It's very nice, basically hand-free. It's really a beautiful camera, very easy to use. Here we have our plate pouring bench. So that technician, if you ever need a specific type of media made, you can just request it in advance, and the technician will autoplate it for you and pour it, and it'll be ready within one day. We have some water baths to keep media stable. We have an ice machine. We have a shaking incubator for overnight cultures. This small room has our pair of autoclaves. These are basically dedicated to our use exclusively, and anybody who has a key to it can just use it whenever they need. Behind this fridge here, we have our liquid nitrogen tank connected to our long-term storage system. All the worm strains that have been made in the lab are kept here in liquid nitrogen across these two machines, and we can always just pull those out with ease if we ever need, and we have a nice spreadsheet so you can look through those. Here we have an array of -80 refrigerators for our long-term storage. This is where we keep our personal C. elegans strains, as well as bacterial clones and our orangular library. Plenty of space, everybody has their own rack, possibly several. And we have one more large shaking incubator here for various specialized uses. On our way out, if we head to the microscope room, I would like to point out that we have a nice, large volume, high-speed centrifuge for our use whenever we need it. This is where we keep our workhorse microscopes, so we're in here frequently. Before I show you the scopes though, I want to point out this bench. This is where we have a dedicated microinjectionist inject the gonad of the worms with whatever strains and whatever constructs we need. So if you need a strain created, you can just ask her in advance to inject for you, and you can give her whatever you want, and that strain will be ready in just a few generations, so it's very helpful to have. Here we have a nice microscope room. We have a postdoc from the neighboring lab. You? Hi. How's it going? I just want to point out a few pieces of equipment in this room. Here we've got a nice Zeiss microscope. It's got an automated stage. It goes up to 100x oil. It's very useful for taking snaps and nice close-ups of pictures or seeing dim objects in the microscope. This one is another workhorse microscope. It is actually a fluorescent dissection microscope. So it's really useful because the working distance between the lens and the stage is quite large. So you can actually pick while you examine worms in fluorescence, and this is really useful for picking out successful crosses or looking at even organelles if they're fluorescently tagged. It's a very useful microscope, and it's got a camera attached. This basically serves the same function. It doesn't have the camera, but it has a really nice 3D depth of field. So across these 3 scopes, this is where we do the majority of our work. We do have a few more specialized scopes, which I'd like to show you in a separate room. This first microscope is a laser exotomy scope, and it can use a high-powered laser to ablate a cell in a pair of crosshairs under in the eyes. So if you want to just target an early developmental progenitor cell, you can actually just hit it with a pulse of laser light and destroy it and see how an animal will develop in the absence. So this is a very useful tool. And one of our favorite microscopes, and one of the most recent additions to our laboratory, is this confocal inverted microscope. This is a really nice scope. It's got an anti-shake table. It has lots of automation, and it is only shared between 2 labs. So it's really, really nice to be able to book time and be able to do time lapses and whatever you need. You'll always have access to a confocal scope nearby. Very useful and very clear, beautiful images. So not only do we have equipment, but we also have a lot of spaces where we can commune and talk and discuss about science. So I'd like to show you our dedicated conference room or seminar room. There are a lot of worm labs here at Rutgers, and during the semester, once a week, one graduate student or one postdoc from one of the labs will give a detailed presentation about their work, and all the labs will congregate in this room and give really productive feedback. Each semester it starts with a potluck lunch, and everybody loves that. And many useful and creative ideas have taken place in this room. It has a dedicated projector, it has whiteboards for chalk talks, it has ample space, so you'll be able to talk to your colleagues at least once a week about interesting and exciting new C. elegans research. Here we have another meeting area, and this is for small group meetings within the lab. And we have many lab groups, such as an exercise group, or the Exifer group, or a group dedicated to eggs and reproduction. And we'll just discuss in our small groups here at this table with our boss fairly regularly. Everybody meets with Monica fairly often. We have small dedicated individual workbenches if you ever just want to get out of the lab and read in a nice quiet space. We have some comfortable chairs, some desk lights, and everything you could possibly need if you want to just read a paper or do some academic work. And last but certainly not least, we have the boss's office. This is where Dr. Monica Driscoll will stay. We lovingly refer to it as the wormhole. Unfortunately, Monica's not here right now, but I can tell you whenever you need to talk to her, you can just give her a quick knock. She'll say, "Come in," and she'll give you whatever advice you need or help you with whatever problems you have. She's very accessible and a wonderful boss. Last but certainly not least, I just want to give you a quick view of our kitchen at the end of the hallway. There are only a few seats, but whenever you want to have lunch or a snack, you can come sit here. We've got a sink, a coffee maker, a microwave, and a relatively clean refrigerator, which is nice and not all too common. Lastly, we have this beautiful view of the trees and outside Bush campus of Rutgers University, and it's a nice place to just sit here and relax every once in a while. So I want to thank you for listening to the tour, and I hope you can come here and see it in person. Once again, I'm Ryan Gwasp. And I want to just thank my cameraman, Guo Chang Wang, a wonderful postdoc in our lab. Thank you.